Hello, and thank you for joining us for Chardan's Virtual Ophthalmology Summit, where the focus of this current installment will be on the rare ophthalmic diseases. My name is Daniel Gatola. I'm one of the senior biotech analysts here at Chardan. It is my pleasure to introduce Shankar Musunuri, CEO of Ocugen. Shankar, welcome.
Thank you. Thank you for having me today, Daniel.
Yes, of course. So the format for this session is a fireside chat. So let's go ahead and get started. First, Shankar, if you could take a few minutes to introduce yourself and Ocugen, including a brief overview of your pipeline to the audience.
Yeah. I'm Shankar Musunuri, Chairman, CEO, and co-founder of Ocugen. Ocugen is a pioneering biotechnology company focused on ophthalmology gene therapies. We are leading ophthalmology gene therapies targeting major blindness diseases with a significant unmet medical needs today, starting with retinitis pigmentosa, Stargardt disease, and dry age-related macular degeneration. Our first program, targeting retinitis pigmentosa, OCU400, is going through phase 3 clinical trial. We're planning to submit our biological licensing applications for approval in US, as well as market authorization in Europe next year. The second program, Stargardt disease, is about to dose patients in our phase 2/3 pivotal confirmatory registration trial. And this will be ready for BLA submission in 2027. The third program, OCU410, targeting dry age-related macular degeneration, late stage, which is known as geographic atrophy, is targeted. It is completing phase 2 clinical trial right now, and we will be initiating phase 3 next year. So bottom line is we have 3 programs targeting major blindness diseases, and we're targeting 3 BLAs in the next 3 years.
Got it. Excellent. Yeah. So as you mentioned, it's you have 3 programs. 2 of those are in more rare indications. The geographic atrophy, of course, is a broader indication. You also have a couple of biologics programs that you'll discuss at a different conference at a later time. So let's focus on your programs for retinitis pigmentosa, or RP, and Stargardt disease. So for these programs, you are using a differentiated gene therapy approach. You refer to it as a modifier gene therapy. Can you explain to us what that modifier gene therapy approach is, and how is it different from the more traditional gene replacement therapy approaches?
Yes. So if you take a retinal space, the modifier genes, how they come in play. Our retinal cells are non-dividing cells, and what you have is what you have the rest of your life. So they also have a specific functional networks within retina. And so there are key functioning genes which are responsible for these key functions, from cell development to metabolism to cell survival. When you have any defect, what we found out, some of these key functioning genes are depressed, any genetic defect. And the missing link is the master regulator, which is a modifier gene or nuclear hormone receptor gene, is also depressed because of the manifestations of the disease caused by defective gene. When you upregulate this modifier gene, the whole network of genes, they restore the expression, restore the homeostasis, also create a healthy environment for cells to survive. That's a very powerful concept because, as I mentioned before, genes don't work in isolation, they work as a network. So if you give traditional gene therapy, first of all, you have to target one gene at a time. That's very complex, right? If you have disease like RP, defects in more than 100 genes can cause the disease. That means you need more than 100 products. It's almost impossible to develop 100 products, target this entire population. Number 2, when you're giving a functioning gene, it's a gene augmentation. You're not really replacing the gene, defective gene. So whatever downward effect of the defective gene, the toxic effects, it's still continuing. So you're giving a functioning gene, you expect to either slow down the disease progression, stabilize. You're assuming the network is okay, but many cases network is not okay. And so sometimes you can question the lasting effect of those traditional gene therapies. So in our case, as I mentioned, our modifier gene acts like a master regulator, restoring homeostasis and the function, as well as creating healthy environment for retinal cells to survive. So that means if we can reset them, potential to survive the rest of life is very high. So based on whatever data we generated in those 4 Nature publication, groundbreaking publications, we are now reproducing the data in humans through all our clinical trials consistently, not only showing the structural, also functional benefits.
Okay, got it. Yeah, thank you, Shankar. Are you aware of any competitors' programs that utilize a similar approach, or maybe some specific examples of gene modifier concept that has shown effect in the clinic or in a lab?
Yeah. I mean, with our therapy, I mean, we don't have any direct competitors. Nobody is using this kind of an approach, holistic approach to really treat the entire network and create this environment for cells to survive. And because of that, we can focus on the entire disease. Like, you need just one product to target all RP, irrespective of what genetic mutation is. Target one product for all Stargardt disease, irrespective of what specific mutation you have, because Stargardt may have 1,200 mutations. So now you come back, there are some optogenetic programs or neuroprotective, and they claim they can also target in gene agnostic way. But their limitation, I think some of them are in late stage, some of them are even getting to BLA filings. So those approaches may benefit severe RP patients. What did severe means? That means these patients are legally blind.
Mhm.
That's beyond 20/200 vision. So they're legally blind patients. They may have some, impact on some impact on their vision, and which is beyond. So those patients may have some benefit using those. Whereas we are targeting early to advanced stage of RP and pediatric patients from 5+. So we're targeting entire RP of more than 100,000 patients with our gene therapy. And optogenetics is on the really, really severe stage of RP patients, which is a small fraction of that. And if there is any impact, time will tell based on how agencies are going to look at those programs, how they're going to benefit the patient and quality of life. So that's the difference. So we don't have direct competition.
Got it. Okay. So let's talk about RP. You mentioned obviously various stages from fairly early where patients are still able to see to all the way the end stage of the diseases where patients are legally blind. Can you tell us more about the retinitis pigmentosa in general? At what age patients begin to manifest symptoms? How quickly they progress? How big is the RP patient population, and what specifically do you expect to be able to target with your approach?
Yeah, I mean, RP in general, defects in our 100 genes can cause RP. The manifestations are depending on specific genotype and epigenetics. Some patients can start the disease progression as early as childhood, and depending on the specific genetic mutation, and manifests itself. And that's why our clinical trial, we're recruiting 5+. We want to treat pediatric patients and stop the disease progression right there. And so that means depending on the genotype, disease can progress anywhere. And some of the patients may not see any phenotype manifestations until late stage, right? I mean, until they're in 50s and they're suddenly saying like, oh my God, I'm losing it. How the disease progresses is similar in most of these patients. They lose their peripheral vision and night vision. Then the central vision is left. Eventually they lose that. And that's why, I mean, there's a patient video on our website too. And I mean, he lost all of his peripheral vision and night vision, and is a central vision left. He wants to hang on to what he has. After our gene therapy, not only is hanging on to what he has, he's getting his peripheral vision back. And so I think that's very important. I think at any stage, and ideally, we want to give it early stage. That's why our clinical trial, if you look at, indication we're targeting, is all RP. And our goal is to get a broad indication for all RP patients from early to late stage, pediatric to adult. So we cover the whole spectrum and syndromic and non-syndromic, because sometimes some clinical phenotypes, maybe you may not be able to diagnose what the genotype specific genetic defect is. So if they think clinically it's RP, we should be able to treat these patients too. There's a fraction of patients who belong to that category. So our goal is to treat broad RP, irrespective of whatever genotype or, syndromic or non-syndromic, broad RP patients. And that's what we're targeting. And that population is 300,000 patients in US and EU, and about 1.6 million or a little bit more global who are struggling with retinitis pigmentosa.
Okay. In terms of the treatment options for RP patients right now, can you walk us through, are there any treatments, or are there any ways for patients to hang on to their visions right now, or that mostly just managing symptoms as much as they can?
Yeah, currently there's only one approved product, Luxturna, that's first gene therapy, and it targets a specific genetic mutation, RP65 gene. So that population for that is less than 1 to 2%, very small population out of the entire RP. And that product got approved. That's the first product many years ago. So that means majority of the population, 98 to 98% of all RP patients across the globe, they don't have anything today. Just like you said, just managing and adjusting their life while they're losing the vision, how to manage their life, sometimes with the help of the family members, and they eventually lose their independence. It's really debilitating disease. And that's why, if we can intervene, get this clinical trial done, and get to the market sooner than later, that's a real hope for these patients. Currently, there's nothing except for that one product, which is very, very small fraction of our RP patients. And many companies, tried to traditional gene therapy approaches, and they were not successful. And so whereas Ocugen, just to remind the timeline, 2022, we started dosing the first patient. And today we're in middle of the phase 3, trying to complete our recruitment, getting ready for BLA submission next year. We're moving very quickly, thanks to my team.
Got it. Okay. And so in terms, as you mentioned, there have been a fair number of programs in the clinic for RP, including X-linked retinitis pigmentosa and others. What learnings from these competitors' programs, if any, maybe in regards to endpoints, study designs, etc., are you implementing, or what other benefit are you gaining from those prior programs as you are advancing OCU400?
Yeah, because the way the disease manifests, losing the peripheral vision and affecting the night vision, typically the approved product uses mobility test. That's what we are also implementing. Also, low light visual acuity, because you lose the night vision first. That's more accurate measure. And also probably you'll be able to measure that sooner than later compared to, best corrected vision, which is under normal light conditions. So those are the two measures even the late stage companies are using, mobility test or LLVA. So what we have done is obviously use the mobility test, which is used in the approved product. And by working with FDA, we also made it more sensitive and specific for phase 3 clinical trial compared to, prior trials, what other people have used. And this test goes from very bright light, 500 lux level, to 0.24 lux levels, which is like 10 different levels. That's the lowest light, call it like a dull moonlight. So what we are measuring here is if I'm able to walk in a brighter light, now with the treatment after 1 year, 12 months measurement, I'm able to walk in a lower light, walking through a maze with the different obstacles. That's a mobility course. And that means the therapy is benefiting me functionally. So that's a measure we're looking at as a primary endpoint. And what we are doing here is we have treated group and untreated control. So we'll be comparing those two. And so that's the first primary measure. The secondary measure is low light visual acuity. And so in phase 1/2, we implemented both, even though primary endpoint is safety. And this year, early year, we released long-term data on low light visual acuity, because mobility test after the clinical trial, most of the gene therapies, you have to follow these patients for 5 years, and so that you can get long-term safety. As a part of that, you can also measure some efficacy measures. And mobility test is very difficult because it's complex. And during the clinical trial, you can do it. And after that, the long-term follow-up, it's relatively easier in doctor's office to measure low light visual acuity. So when we got the data this year on evaluable subjects from phase 1/2 after 2 years, it's compelling. And we got a p-value of 0.005 in these patients that included, gene agnostic patients, rhodopsin. And so that's a good measure we are using. And so that's why in phase 3, we're following mobility test, which is, we call it luminescence-dependent navigation assessment. It's proprietary to Ocugen. Working with FDA, we created that. And then LLVA, low light visual acuity, secondary measure.
Okay, got it. All right. Yeah, we'll talk about more about your phase 3 design and the progress. I want to spend a few minutes on the phase 1/2 study in RP. So you completed that. You shared the data. Before you give us the overview of that, can you briefly give us an overview of the design and what patient baseline was enrolled in the study?
In the phase 1/2, we're looking, primary endpoint is safety. So you have to do low, mid, and high dose escalation. And in the dose expansion phase, we included more patients. The total we used for retinitis pigmentosa is 18 patients. And so what we did, based on that design, based on the data, number one, the safety is good. That means it's safe and tolerable. We don't have any severe adverse events related to our gene therapy. That's number one. Number two, we also establish all 3 doses are safe. And also the way the modifier genes work, we believe once you hit certain threshold, you may not see any dose response. So we didn't see any dose response. As a result of that, we picked a dose between mid and high before we go into phase 3. That's because in phase 3, we're going into pediatric population. So we took a dose below high dose and above mid dose. So that's a target. So we can take single dose, move into phase 3 based on that.
Mm-hmm. Okay. And
also one question, Daniel, I think you asked,
sure,
what gene agnostic. So that also included multiple genotypes, defects, NR2E3 related, also rhodopsin. Rhodopsin is a big group we used, majority of the patients, because it's a tough group. Most of the rhodopsin mutations are autosomal dominant. That means if you use a traditional gene therapy, there's a potential for gain of function or gain of dysfunction. So that's why traditional gene therapy companies didn't attempt to work on it, because this group is a big group. It's a 10 to 12% of entire RP. So why didn't companies attempt to go there? And so there is a reason for that. There's a potential safety. So in those patients also we recruited, and that therapy is safe and tolerable in those patients and showed effectiveness based on phase 1/2 results.
Okay, got it. So in terms of you alluded that you didn't see clear dose response. Was that between all 3 doses, or were you able to hit this threshold with dose 2 and then you didn't see much difference between dose level 2 and 3?
Yeah, I mean, we didn't see much difference. I think the data looks good for all 3 doses. And obviously, it's good to take, if it's safe, it's always good to take the higher dose than lower dose. So we picked something in between mid and high.
Gotcha. Okay. And in terms of durability, so you're obviously following these patients for extended periods of time. As you saw improvement, were patients able to hang on to that improvement? Did you see any continued improvement over a longer period of time, or did any of the patients see any worsening?
No, this is a great question. So when we looked at the evaluable subjects after 2 years, we saw very good effect from treated versus untreated eyes, looking at low light visual acuity with a p-value of 0.005, statistically superior, clinically meaningful, And also, we looked into all the subjects have either stabilized or improved on the treated eyes. So we went back and looked at the same group at 1 year, from 1 year to 2 year, even 1 year met a p-value of less than 0.25. From 1 year to 2 year, we saw further improvement. So that's good. I mean, once again, going back to concept of modifier genes, creating this healthy environment. I mean, these genes may take, sometimes because it has to work on the entire network and the way the gene is expressed and downward effects, it may take some time. I mean, you may see effects in, 6 months, then slowly you'll start seeing further effects and improvement. That's what we have observed in phase 1, 2. I mean, 1 year is good, 2 year looks better based on LLVA. So that's good. It's durable and safe and getting a long-term data.
Okay, got it. And so in terms of dynamics of response, you mentioned that because it's not a direct gene replacement therapy, and it has to essentially work its way through a cascade, that it may take potentially longer. When are patients first seeing efficacy response? Is it 3 months, 6 months, or is there a median time period when they actually start seeing the response?
I think 3 months is little soon, but within 6 months, I think 6 months you'll start seeing it, 3 to 6 months range, but 6 months you'll start seeing it. And then 6 to 9 months, slowly you'll see more consistent response, and it's further improving. So you need I believe in about 6 months, because we don't measure in the middle, like we do 3, 6, 9, and 12 months. And so those are the measurements typically we do in these clinical trials. So, I mean, I think 6 months is a good measure.
Okay, makes sense. If you were to summarize the phase 1, 2, or kind of put your opinion about phase 1, 2, what part of those data do you think is most exciting to you as a company and to retinal community as you speak to them?
I mean, number one, first and foremost, the retinal community, there's nothing for them except for one product, which is less than 1 to 2% of the population. And that first product was ever approved in 2017. Today, it's 2025. Many companies try to work on traditional gene therapies. There's nothing out there in the market to date. So looking at phase 1, 2 results, we showed, number one, the product is safe. Number two, looking at mobility test, we showed the data, how treated and untreated eyes it's impacting. Majority of the patients are either significant number stabilizing or improving with the mobility test. And we also showed long-term data on LLVA, which is a very good functional measure. So also gene agnostic approach, taking rhodopsin, which is a tough autosomal dominant mutations, showing effect in those patients. That's important. Why? If you have a gene therapy and showing gene agnostic effect, that means demonstrating this can work in potentially all of RP. That's a big thing for retinal community. So the hope is, that hope is alive and we'll be there shortly.
Gotcha. Okay. So for phase 3, as you mentioned, you're looking at patients who have both the RO mutations as well as gene agnostic, right? You split those into separate groups. In phase 1, 2, you also looked at all of these patients. Did you see any difference in response between patients with RO mutations and others?
I mean, in phase 1, 2, obviously we had a limited genotypes, right? We used majority of the patients are RO or NR2E3, and NR2E3 auto. I mean, there is the dominant and recessive groups, and recessive groups obviously I think fared a little better than NR2 itself, the genetic defect, but rhodopsin patients did really well. So I think in phase 3 we have a lot more mutations we are covering. We're covering all the major types. Which has a high prevalence, including, and so our gene therapy is open to, patients with the XLRP, XLRS, including some of other mutations, which are in large numbers. People are looking for therapies. We include all those patients. Our goal is to cover all the most prevalent groups in RP, and those groups are being covered in phase 3. So we truly can show the gene agnostic approach, mixing all these groups in RP. So going after the broad indication.
Mm-hmm. Okay, got it. So let's talk about that phase 3. So it's called Limelight study, I believe.
Yes.
The enrollment is ongoing. Can you quickly summarize the study design? How many patients total are you looking at, and the overall groups and number of patients per
Yeah.
group?
Yeah. It has got two arms. One is rhodopsin, because we had good data in rhodopsin arm. So in phase 3, there's a separate arm, 75 patients in that, 2-to-1 ratio, 50 in treated and 25 in untreated group. There's another arm, gene agnostic. All other mutations are in that, 50 in treated, 25 in untreated. So it's 2-to-1 ratio. It's a 1-year duration. And the mobility test, which is LDNA, is a primary measure at 1 year. We're going to compare treated versus untreated. And similarly, the secondary outcome is low light visual acuity, and between the treated and untreated. And so those are the endpoints.
Okay, and this study is enrolling both adult and pediatric patients, correct?
Yes. Yes.
Okay, got it. So as you mentioned, the primary endpoint is luminance-dependent navigation assessment, or LDNA. What's the difference between this test, LDNA, and other previously used mobility tests?
I mean, this test is more sensitive and specific. I mean, in all other tests in the past, they must have used 7 or 8 levels. We actually extended it to 10 levels. Actually, on our corporate deck, you can see it. And some of the levels are also evened out based on FDA input for phase 3. And so the technology is very sophisticated now. And in the past, they stopped mostly at 1 lux level, like a moonlight. Now we can go down to 0.04. Why that's important for patients? Because some of the patients in a clinical trial setting, they may be improving with gene therapy, but you have to show in a quantitative way. How are you going to show it? And if you're already at 1 lux level, even though you may be showing through other measurements, your function is improving, you feel it as a patient, but I cannot show it in the clinical trial. So that's why, thanks to FDA, they allowed us to go to 0.04 levels. So in between 1 and 0.04, there are multiple levels there. So at least we can show, even if you're at 1 lux level, moonlight, that's all you can see. You cannot, go below that. Now with the therapy, we can show you can further improve. If you're not able to do that, such sensitive level going to 0.04, even though you're improving, I cannot show the effect. So you have the ceiling effect, you call it. So you cannot be enrolled in the clinical trial. So that's a criteria. Number one, first thing is we screen patients, ability to improve multiple levels, like 2 levels at least, during course of 1 year. Otherwise, they won't be qualified for the test. And so that's an improvement we did. And also, we have sophisticated calibration in a validated test. And so that's more specific and sensitive, and also goes to from 400 level to 500 lux level on the high end, very bright light, to on the low end going down to 1 lux level to 0.04. Those are all the changes compared to prior mobility tests people used to get approval.
Gotcha. Okay, I understand. So for XLRP, there have been discussions recently about what primary endpoint might be more suitable, either a mobility test or LLVA, or low luminance visual acuity, which you're using as a secondary endpoint. Can you tell us about your thoughts on this, and maybe if you could share how your discussions with the FDA went that led to your agreement to use LDNA as a primary endpoint instead of LLVA?
Yeah, I mean, both the measures are good. I mean, obviously, when a product gets approved, precedence makes a big difference with any regulatory agencies. If you follow the path, they use mobility test because there's a justification. That's a good functional measure. How in real life and quality of life, functional improvement. I mean, that simulates, right? Walking through a maze. And LLVA is a good test. There's also a lot of, I mean, there are some, LLVA definitely is a good test. But, defining what is clinically meaningful. If you talk to most of the retinal surgeons and experts in the field, anything you improve above 5 letters, because single line is 5 letters, if you that reading chart, right? Anything above 5, below 5 is kind of a noise. Anything above 5 is meaningful. So working with the regulators on, and historically, they were implementing the 15-letter number, right? Coming from different diseases, which are not relevant to retinal degenerative diseases. Some of the diseases, it could be wet AMD. And when you have diseases like that, where there is leakage, which is blurring your vision, if I can dry it out with therapies, suddenly the vision improves. In this case, irrespective of what diseases, all the diseases work, there are retinal degenerative diseases. Your retina, in Stargardt or GA, your central vision is degenerating. That means you got atrophy that's growing. In RP, your peripheral vision, those cells are getting, inactive. And so in these cases, what you're measuring is you should have a clinical benefit for the patient. What does it mean? If you can show by treatment, my treatment is, either stabilizing or improving compared to untreated. Untreated controls are losing, and they're not, their vision is going down, and treatment is stabilizing or improving. If you can show clinically meaningful with any of these measures and benefit, because it's a significant unmet medical need and it's safe, those products should get approved because there is nothing for these patients. So I think I'm hoping regulators are going to closely look at it and not to use the historic 15-letter came from different disease and applying to retinal degenerative diseases. I think there is a lot of debate about it. And I'm hoping, they're going to seriously look at it and define what is clinically meaningful. So that's why mobility tests, at least, when Spark did the clinical trial, they show a benefit treated versus untreated arm and show statistical superiority, and they got the approval. And so at least there's a precedence, and that's why we followed it. Can LLVA be used? Yeah, LLVA is a great measure for all the programs we're working on, because most of these diseases, any disease, even we as a normal, if you think you have healthy retina, as you age, you have low light vision issues first before bright light, right? So that's why potentially the low light measurements you can measure accurately within a shorter time, like 1 year or 2 years. Maybe the brighter light effects you can measure in longer time. So in a clinical trial, especially there's an unmet medical need, you want to do the measures which you can measure meaningfully in 1 year to show the difference. If you run like a longer trials, yeah, you can look into other endpoints. So that's important. There's a unmet medical need, and how do you get, if there's a benefit to patient, to the market sooner than later? These are the measures. I think they both are good measures, and obviously LLVA from regulators' perspective needs some adjustment.
Gotcha. Okay, yeah, makes sense. For Octio 400, you recently announced that you signed a term sheet for the license for RP in Korea. Can you tell us a little bit about that partnership and what does it mean for Ocugen?
Yeah, absolutely. It's a very good, solid regional partnership. We really want to maximize our reach. If you look at our mission, not only we want to take this game-changing gene therapies to the market in the next 3 years, also work even harder to provide market access to patients who need them. So that mission already started. So this regional partnership perfectly fits into that. Having a good players in that region can maximize the potential for patient outreach. That's number one. Also holding off the bigger markets for later stage so that, we can also take care of the shareholder value. So we had to balance those. And so I think our goal is, reach to as many patients as possible with our therapies. I think these regional partnerships can actually maximize that. So that fits into that, and we're happy with that. And we'll continue to discuss with potential partners in multiple regions.
Got it. Okay. And one last question on RP, just to kind of bring it all together. What are the potential timelines for the next steps?
I think the next steps are, closing down our recruitment and also working with FDA. Because we have a regenerative medicine advanced therapy designation, we are eligible to, initiate a rolling submission, because that also helps with agencies to, and so we can we are completing our process validations from CMC perspective, and we have a robust process and will continue to do well. We'll be completing those runs. So the CMC part is going to be done. Non-clinical, clinical, CMC, we can actually roll those submissions through the rolling submission earlier. We're waiting for clinical data. When the clinical comes in, that will be the last section. That will speed up with the regulators, and also it will benefit them too, having other sections start reviewing them. And once the clinical is dropped, it's 6 months from there. So we're on target from CMC perspective. We're doing well with the recruitment. And so we're hoping we're on target for filing our BLA next year. In addition to that, through ATMP process, European Medicines Agency gave us a centralized procedure, which makes it easier. And we're closely working with them too to complete that market authorization filing next year in Europe too, in parallel.
Got it. Okay, got it. Excellent. Let's spend a couple of minutes and talk about your Stargardt program, OCU410ST. Maybe in a minute or so, if you can give us a quick overview of that program and where you're at, the current status.
Yes. Yeah. Stargardt program, we announced FDA cleared our IND amendment to go into phase 2/3 registration trial. So this is a phase 2/3 pivotal confirmatory trial. So what we did, instead of doing phase 2 and phase 3 in a sequential way, that would have taken additional 2 to 3 years and significant resources and capital. And thanks to FDA, with the promising data coming out of phase 1 safety and efficacy signals, we're able to work with them and convert our phase 2 into phase 2/3 registration trial. So we're about to dose patients, and first and foremost, we're going to continue to find these patients. It's 51 patients. It's 2-to-1 ratio, and 34 in treated group, and 17 in untreated arm, and SSL blinded study. And we also created an adaptive design. After 8 months, we can look at it. And so if you want to adjust or extend the time, because some of the rare diseases, you're seeing the effect, you may need to go longer time to differentiate treated versus untreated, which benefits the company to lower further risk from the program. FDA liked it, so we created adaptive design. So the clinical trial is 51 patients. At 8 months time point, we'll take a look and see if any adjustments are needed. The trial goes on for 12 months. That's a measure. Lesion size, which is structural, is a primary endpoint. And secondary endpoints include low light visual acuity and BCVA.
Gotcha. Okay. And as you mentioned, Shankar, you've had fairly recent interaction and alignment with the FDA to convert this into a single pivotal registrational study. How did these discussions come about, and more generally, what does that outcome tell you about the agency's position on the rare IRD space?
Agency's position, I mean, so far with our interactions with the agency, very, very supportive and positive. And when there is a significant unmet medical need, and they understand it, and also gene therapies, like our gene therapies, are safe. In all our programs to date, we dose so many patients, and we don't have any severe adverse events related to our gene therapy. That's important. If the programs, if the safety looks good and it's benefiting patients, I think they see it. And I mean, our interactions and everything to date is very positive based on the Stargardt approach we have taken, and how promptly they responded to our needs, and made us move forward very quickly into our registration trial. And thanks to them.
Got it. Okay, makes sense. In terms of your CMC strategy, I believe your plan at first is to work with CDMOs, right, for both 400 and 410 ST, but you also have internal manufacturing capabilities. When do you expect those to be fully functional, to be fully online, and further in the future, do you expect to start manufacturing these therapies yourself?
Yeah, I mean, once again, we established a very good process from clinical, phase-wise, what is needed from quality perspective, really a robust strategy we built on, focusing on process consistency, product compatibility, and analytical rigor from clinical transitioning into commercialization, and also using platform technology concept. You got the same plasmid backbone, cell banking, scalable, HEK 293 base production, and also downstream purification. So a lot of these factors are built in. And so what we learned from OCU400 can be implemented for OCU410ST and very quickly. So what we have done is, as I mentioned before, we're in great shape with the first program OCU400 and process qualification runs supportive BLA. And Stargardt, we already actually have a commercial scale process for phase 1. And so there's no change. And the robust process is being moved into phase 2, 3. And so that's important. So from CMC perspective, we're in a great shape.
Okay, understand. Finally, just a couple of general questions in terms of the company's current cash runway and what assumptions go into that runway.
Daniel, I think one question I didn't answer. I just want to finish that on the manufacturing.
Sure,
We do have our US manufacturing. Currently, we're producing from our ex-US partners for launching OCU400. Our facilities in our backyard, Malvern, Pennsylvania, they're built. They need some more work. Our goal is, when the product is launched in 27, in the next 2-3 years, get those facilities up and running and add those facilities to our gene therapy. So we built enough capacity, and once these upgrades are made and get ready for GMP production, our goal is to, once we get the first approval and add the US site, and we will be able to support all our US supply from our Malvern site. And so, our goal is to continuously work on this in the next 2-3 years. And when you get the first product approved, we'll be able to add the second site and also rely on our US manufacturing in US from our Malvern site for all our gene therapies. That's our goal. And
gotcha.
Coming back to cash runway, where we are, and our cash runway goes into first quarter next year, as we mentioned to the market consistently. And our burn is a very efficiently run company. We have 12 million a quarter, about 50 million a year. And so we're carefully watching it. And as we mentioned, we announced a regional partnership deal. We're looking at those BD deals right now. And based on where we are in the next few months, we're also having a significant inflection point coming up in the next couple of months. We're going to release interim data from our dry AMD trial. That's a 54-patient trial. So midway, at least 50% of the patients reaching midway point. It's a 12-month monitoring, about 6 months. And we already saw very promising data from phase 1, structural as well as functional benefits to patients. And so we're going to announce that, and we're going to watch that and see where we are based on this BD, potential upfront and milestones. We're going to balance out. If necessary, our goal is to extend the runway into 2027.
Okay, got it. Makes sense. Awesome. All right, I think that's all that I have, Shankar. Again, thank you very much for joining. It's been a pleasure talking to you.
Thank you for having me again.